Well, hello, everyone, and welcome back to Science in the Age of COVID-19. Today, we are super happy to have Dr. Peter Palese from Mount Sinai. He is a professor and actually chair of the Department of Microbiology. If I had to pick one department in the world that has made the most progress on COVID, I would almost certainly pick this department. We have had Florian Kramer from his department on the series 3 times, and Ben Tanuver either 2 or 3 times. I can't remember which. Peter's lab works mostly on RNA viruses, historically influenza. Lots of milestones in Peter's career. He reconstructed the pandemic, the 1918 pandemic strain, helped establish roles for hemagglutinin and neuraminidase, host factors, role of the mitochondrion in the pathological cycle, many things. Today, he will tell us about progress towards a universal flu vaccine. So maybe we wouldn't have to get boosters every year and have them fail a lot. And he will talk about should be a stable, low-cost COVID vaccine. So Peter, take it away.
Thank you very much, Lauren. You almost said it all. We really want to have a influenza virus vaccine which we don't have to take every year, but which would last maybe 20 years or even a lifetime. And then we have also an attempt to make a cheap SARS coronavirus 2 vaccine, which is based on a vector which is not adenovirus, as you all know, but rather a Newcastle disease virus, and we hope that this particular vaccine may be have certain advantages. Now, let's start with influenza viruses. This is an electromicrograph, and the outside of this virus consists of the hemagglutinin, in this case subtype 1, H1, and the neuraminidase. And on the inside, we have these black spots which are made up, consist of the 8 RNA segments, 7 around and 1 in the middle. And these 8 RNA segments code for the genetic information of the virus. Now, the important thing is that the hemagglutinin and the neuraminidase are those proteins which are recognized by our immune system. And if we change the hemagglutinin and the neuraminidase dramatically, as has happened over the last 100 years, we get what is referred to as a pandemic or an influenza virus epidemic which goes around the world. And if you look at this slide here, we start with 1918. That's a famous pandemic of 1918/1919. And then we go on until 1957. And you can see here the color changes. The color changes indicating that the surface of the virus, particularly the hemagglutinin, changes sufficiently so that after 2 or 3 years, the virus can reinfect us and can also cause disease. So if I get infected in 21, I can be reinfected with a slightly different strain in 2023, 24, and that makes it very difficult in terms of vaccines because one has to change the formulation of the vaccine, not every year, but as you will see it quite often. When we have in 1957, we have a change in the hemagglutinin from the H1 in 1918 to the H2, and that resulted in one pandemic. 1968, another one when we had from H2 to H3, and then in 2009 we had again a different pandemic, a different H1, different enough from the other one so that we got again a pandemic. And then we have also influenza B viruses, which are constantly circulating in the human population. Fortunately, we do not have these pandemics and these different hemagglutinin subtypes among the influenza B viruses. And so it's a little bit easier these strains do not vary as much. Now, this is a slide which I always like to show because it illustrates a dip of 11 years in 1918, 1919 of about in the life expectancy, 11 years in the US. And this is for both male and female. And if you look at 1900, the average life expectancy was about 47 years. Now we have a baby girl born in 2021 is way over 80. So we have a straight curve, but we have this major dip, which is a result of the pandemic of 1918, 1919, where about 750,000 people died in the US alone. We had only about 100 million people as compared to the 340 now. And worldwide, we assume that about 100 million people died, including tremendous number of deaths in Africa, in Asia, etc. So this is one slide which many of us have seen many, many times. It shows Camp Funston in 1918, and you can see that these were all people who were sick of influenza, and it was not known at the time that the disease was caused by an aerosol, by a virus in the air, but you can imagine that this was not the most healthy environment. And people, if they were not sick before, they were certainly sick when they were in these hangars, really, in terms of many people died, obviously, and they were taken care of by military personnel, but it was a major, major problem. As Lauren has mentioned, we were able to reconstruct this 1918 virus in the laboratory, and this is sort of a paper which is now 15 years old, and we were able to characterize the 1918 Spanish influenza pandemic by reconstructing it in the laboratory, and this really gave us a lot of insight in terms of how this virus behaves. It's certainly the most virulent human influenza virus with which I have ever worked with. And Adolfo Garcia Sastre is the last author on this paper, and it is based on the sequence by Jeffrey Taubenberger, who got it from material which was obtained by people who died of the 1918 flu. And Terry Tampe is the first author, and he rescued this first virus in a BSL-4 facility because at the CDC, because we didn't know how dangerous this virus would be. But very briefly, we found that it could be inhibited by vaccines. We made vaccines against it. We showed that the 1918 virus was also sensitive to the antivirals which we have now. So even if the virus would appear today, we would not have the same problem as in 1918. So the influenza virus vaccine is unique in the sense that is the only one which has to be given and re-given annually, because the formulations which are hopefully protecting against the next season of vaccine changing constantly, and this is a real problem. All the other vaccines are lasting much longer. I mean, smallpox vaccine can really last a lifetime. Measles vaccine is a very good vaccine. The one which gives us really problems is the influenza virus vaccine, and therefore, and because the virus is constantly changing. Now, this is a slide which Dr. Fauci many times shows, and it is very interesting that we have a lot of cases, and this is 2010 to 2016 in the country. These are US numbers, and then we have the number of hospitalizations, but also the number of deaths can be pretty high. As you can see, it can go up to 80,000 per year as a result of a seasonal influenza. So this is something which is something we should understand, that this is a yearly burden and can be quite dramatic in terms of some years are worse than others, particularly when the vaccine isn't really matching with the circulating strain. But you can see that this is a recurring problem in terms of influenza virus infections. Now, if we just look at the last 20 years, we have 4 different strains circulating. Again, the color change indicates that we have these antigenic changes from year to year, but we have one H1, which is the pandemic H1N1, which appeared in 2009. We have one H3N2 virus, the brown one, and then we have actually 2 different lineages. So we have in 2021, we have actually 4 different influenza virus strains circulating. We have 2 influenza A viruses, the purple and the brownish one, and then the yellow and the orange one for influenza B viruses. And what is important is that we have in the present vaccine for the 2021-2022 season, we have 4 different strains, as I said, 2A and 2B strains. Green means they had to be changed from last year, and black is the only one which was not changed from last year's 2020 to 2021 season. So you can see in this case we have 3 out of 4 changes, and that means companies, manufacturers have a hard time every year to adjust the composition of the vaccines. And we have not only these seasonal changes, but we also have these pandemic changes, which I mentioned before, 1918, 1957, 1968, 2009. And that is because we have these different subtypes of influenza A viruses. We have 18 different ones. So far, we have in humans only in the last 100 years, H1, H2, and then H3. But these other hemagglutinin subtypes are actually in animals, in bats, in birds, in horses, in pigs, even in whales, reindeer. So they are ubiquitous, they're everywhere, and unfortunately, some of these influenza viruses can jump into humans. So this is sort of the unfortunate situation which we have, namely that we have this variation, both in terms of antigenic drift from one year to the other, but also occasionally every 10, 20, 40 years, we have these new H hemagglutinin subtypes appearing, which result in pandemic influenza. Now, what can we do in terms of a universal influenza virus vaccine? And let me just show this one slide here, the left side, which shows the hemagglutinin spike, which has a stalk, which is green, and down here is the membrane of the virus, and up here is the head. And the red head is something which is more immunogenic than the stalk, and it varies dramatically. So if we would be able to make a vaccine which induces an immune response against the stalk and not against the head, we would be in a position to make a universal influenza virus vaccine, because the stalk doesn't really change over 10, 20 years. And that's actually what we try to do. So without going into details, really, we showed that by transposon mutagenesis, we can identify the part of the hemagglutinin which is subject to change because of the antibodies, and we were able to really show that the stalk is much more conserved. And that's really what we are doing if we make these universal influenza virus vaccines. You all are aware, unfortunately, the influenza virus vaccines which are presently produced are not optimal. They vary in their efficacy from 20 to 60, 70%, but not higher. They belong to 3 types: the inactivated, not inactive. So that means the virus is grown up, and then it is with formaldehyde or beta-propionlactone, it's inactivated and then injected. There are live attenuated vaccines which are sprayed into the nose. And then we have also recombinant vaccines, meaning the hemagglutinin, just the hemagglutinin is made usually in baculovirus, in insect cells, baculovirus carried hemagglutinin genes, and these three different influenza virus vaccines are actually FDA approved, and we are using them. Unfortunately, I said they are not as frequently used, and I think the compliance is not as good as we would hope that these vaccines have a better acceptance in the population, because even though one might get some influenza virus symptoms, the overall symptoms are much lower and much less in a person who was vaccinated as compared to one who was not. Nevertheless, we want to make something which is even better than what we have, and that is the universal influenza virus vaccine. Now, I show again this hemagglutinin, and green is the stalk, which is much more conserved. And what we have done is, and we is a collaboration of the three groups of Florian Kramer, Garcia Sostre, and myself. And we have made what we call chimeric hemagglutinin constructs, chimeric CH8/1. So the /1 stands for the stalk is the type 1, serotype 1, which we have circulating at the present time. And then we put on the top, not an H1, but an H8. We meaning people have never experienced that. And therefore, when we vaccinate with this, we make some antibodies against the stalk, because we have some memory cells, B cells against the stalk, but it is not enough to be really neutralizing in terms of the new viruses. And the H8 head we have never seen, so we make very little immune response. And then we boost with a second vaccine, which is a chimeric H5/1, so it's identical to the hemagglutinin on the left side, but it has also a conserved stalk, but it has an H5, again, a hemagglutinin, which we humans have never seen. And so what that means is that we are inducing, after having given these two chimeric hemagglutinin vaccines, we have induced the immune response to be enhanced by, in terms of the H1 response, but the H8 and the H5 and the tip of the hemagglutinin is sort of ignored. And this is really the trick we believe will be successful. We can protect all the mice in the world against all influenza viruses doing those kinds of vaccinations. And the problem really is it as good in humans as it is in ferrets and in mice, which are the regular animals which we are using in terms of studying influenza virus vaccines. So we have many data showing that now these kind of vaccines protect not only against different H1s, but H2, but also against the other different influenza viruses which may appear. And we are very hopeful that this is a vaccine which will protect against all influenza virus strains. Past, that's what we can check, but also in the future. So once more, when we look at the influenza virus vaccine, we make one where we have the hemagglutinin, which is shown here in terms of a trimer with a head and a stalk. We change the head such a way that we silence the head. We use a hemagglutinin head, which we have never seen, but the stalk we have seen, and also the neuraminidase, and therefore we make an immune response against this, and we are able to really protect all the hamsters, all the different animals we have tried against an influenza virus challenge. So let me sort of skip some of the data we have also with ferrets. They are very, very good. And this is the ferret. It's actually not a very nice animal. They can bite, and they are really, they're pretty mean animals, but they are very good in terms of replicating influenza virus in their throat. And so what we have done is we have vaccinated with these chimeric constructs, and we showed, and they didn't show the data really because this would be quite time consuming, and we have quite some interesting and compelling papers on this. We can protect mice from the same hemagglutinin variations of the theme, but also from different hemagglutinins in terms of heterosubtypic challenge. And we can do this also in ferrets. And we have been able to show that this protection is antibody mediated many times through ADCC, antibody-dependent cell cytotoxicity, and that is the mechanism, less so by direct binding to the tip, because the tip is really different, but we are inducing an immune response against the stalk, which is very cross-reactive with all kinds of different influenza viruses. And we have a Nature Medicine paper, which appeared in January of this year, where we show in humans a chimeric hemagglutinin-based universal influenza virus vaccine induces broad and long-lasting immunity in a random placebo-controlled phase 1 trial. So this is was financed by the Bill and Melinda Gates Foundation, as well as by GSK. We used the adjuvant from GSK, which is very, very good. So this was a very successful trial, and we are now doing additional phase 1 trials with influenza B viruses and with the group 2 hemagglutinins, and hopefully by mixing those 3 stalks, group 1, group 2, and influenza B viruses, we will be able to have a effective universal influenza virus vaccine. Now, again, summary, a universal influenza virus vaccine, we do this by reducing the immunodominance of the hemagglutinin head, the tip, and increase the immunogenicity of the stalk, and also that of the neuraminidase, because that is also more conserved than the hemagglutinin head. And as I said, we are the mechanism of action is antibody mediated, and it is involving the activation of FC receptor effector functions. So it is a very effective neutralization of different influenza viruses. Now, we are doing this, as I said, in a collaboration, Adolfo Garcia-Sostre, Florian Kramer, and we have other very effective collaborators, Jeff Ravitch in terms of the mechanism, and Patrick Wilson, who is also collaborating on many of these experiments which we have done. And we have, as I said, we were supported by the Bill and Melinda Gates Foundation, as well as GlaxoSmithKline, and for most support really comes from the NIH and philanthropy money which we get through Mount Sinai. And then we have a very effective group of postdoctoral fellows and students who have been with us for many years developing this universal influenza virus vaccine. Now, this leads us to the second part, and that is the COVID-19 vaccine. Let me say right from the beginning, we all know we have these variants and we are concerned about this. So we are not there yet in terms of a universal coronavirus vaccine. And I think only time will tell whether the variation is really as much as we see with influenza virus. At this point, I think the jury is still out whether the degree of antigenic variants is such that we have to really think about making and remaking the vaccine every season, but time will tell. We all know this is a real problem, and this is the virus causing the real problem. It's called coronavirus, and it has like a crown. It has these sort of strangely formed spikes at the surface, and that is the famous SARS coronavirus 2 spike. And we make vaccines. Basically everyone makes vaccines using that as the antigen to protect against SARS coronavirus 2. Now, as of last week, we had 150 million cases worldwide, over 3 million deaths, terrible degree of really a global epidemic, a very terrific number of deaths in people who have long COVID and all of that. And we really can only succeed if we have a good vaccine. Now, the COVID-19 in the United States, again, from last week, we have close to 600,000 deaths and easily more than 30 million cases. So this is really something which is terrible. If we look at the New York City, we see that we have, in essence, 3 different waves. We had the first wave around March, April of last year, and then we had a fairly mild summer, fortunately, only to be hit again in January of this year, and then it is sort of lingering on. Fortunately, it is coming down quite dramatically right now, and we just hope that the number of vaccinations will go up and that we will get a herd immunity by vaccination as well as by people who have been infected by the real thing. Now, we have heard a lot about influenza virus vaccines, but we have also heard about a lot about COVID-19 vaccines. And they are number 1 and 2 are really the mRNA vaccines, which are being produced by Pfizer and Moderna, and also have received at least partial approval by the FDA. We have also the viral vector vaccines in terms of a different sort, and most of them are actually the viral vector is adenovirus, and there is the famous AstraZeneca one, CanSino, which is a Chinese one. The Sputnik 5 is a Russian one, and Johnson & Johnson is a US one. So we have 4 of these adenovirus-driven vector vaccines, which are very effective. And then we have also inactivated vaccine, and these are mostly Chinese, in which the virus is being grown up and then inactivated either by beta-propiolactone or by some other alkylating reagents, such as it could also be UV light and also X-rays. So there are many ways of inactivating these vaccines, and the most common one is really by beta-propiolactone at this point. And then we have also recombinant protein vaccines, and the Novavax, which is not yet FDA approved, but that is made in baculovirus and is actually it makes, or the hemagglutinin is being expressed in these baculovirus infected cells, and they form some aggregates which are quite immunogenic. So there are a lot of these different approaches, and ours is one which is similar to the adenovirus vectors, but is not using adenovirus as a vector, but rather Newcastle disease virus as the vector, and we believe it has certain advantages over the AstraZeneca and the Johnson & Johnson one, in that sense that they are not using a human adenovirus or a adenovirus which humans have seen, and also it is not, we do not use human cells to grow this up, but we are using chicken cells, embryonic chicken eggs, in terms of producing the vaccine. So I have already said we use Newcastle disease virus, and this is an avian virus, a chicken virus, and that causes disease in virulent strains cause disease in chicken, but we are using a lentogenic NDV strain, which is used in chicken as a vaccine and do not cause any disease in chicken. And there are different strains which are used all over the world, Lasoda, Hitchner, and Fuller vaccines. So these are specific strains. We use the Lasoda one to make an NDV-vectored COVID-19 vaccine. And just to sort of stress what the advantages are, it is a safe carrier. It's a non-human pathogen, so it does not cause any disease. And also, it has been used as an oncolytic agent in humans, several hundred cases, several hundred patients have been given Newcastle disease virus as an oncolytic agent and has a very good very good safety profile. The growth profile is also very good. It replicates the very high titers in embryonic eggs and actually grows even better than influenza viruses, and therefore the cost of production is very, very low. We found that the genome of Newcastle disease virus tolerates the insertion of a large trans gene, and this is the SARS coronavirus S protein, which we express in Newcastle disease virus. And as I mentioned, it is already because it can be grown in embryonic chicken eggs, it's a low cost, and this can be easily scaled up and would be very effective in low and middle income countries. And one other important point is that in contrast to the mRNA vaccines by Pfizer and by Moderna, it does not require very low temperatures for refrigeration. We can use 2 to 4 degrees centigrade. That's a home refrigeration temperature. And there is basically no cold chain because regular refrigeration temperatures are very easy to obtain. So this is very briefly what these advantages are. And let me just show you. So we have the Newcastle disease virus in black here on the upper left corner. And we have introduced here the blue is the SARS coronavirus S protein spike. We have removed the polybasic cleavage site, and we have introduced for stabilization 6 prolines. And this was discovered by Jason McClellan, who gave a talk 2 weeks ago. He was able to find 4 respiratory syncytial viruses, as well as for these different coronaviruses, that prolines can stabilize the pre-fusion state of the protein, and therefore is very, very stable. And this is important because we can make viruses now, NDV viruses, which express in blue the SARS coronavirus 2 spike. So this is really was possible through the methodology which Jason McClellan from Austin, Texas, developed, that one can get very stable viruses with the SARS coronavirus 2 spike inserted. And we have live and inactivated platforms. We can give it through the nose as a live one, or we can inactivate. We can use also intramuscular live or inactivated inactivated, and therefore induce an effective immune response against the spike protein. So this is something which we have developed over the last several months, and I can show here a polyacrylamide gel, which is, we call it the HexaPro S vaccine. It's abundantly expressed by Newcastle disease virus. And on the right polyacrylamide gel, we can see in the lane 1, we see the molecular weight. And then we have a preparation passage 1, passage 2, 3, and 4. And we can see that the band in blue is the spike protein, whereas the polyacrylamide gel on the left side doesn't show this band and just the L. And this shows us that the hexaproline S is very effectively expressed on the surface of the virus and almost as much as the HN on the virus particle. So this is very effective in terms of how the virus presents the spike protein, and we are able to make a very effective vaccine. Particularly, we have very compelling data in terms of an inactivated vector. So the inactivated hexapro is immunogenic at low doses and confers protection in the mouse and the hamster model. We can also use adjuvants, and it can be produced at very low cost. And we can also make all of these variants which have been described, the South African variant, the British variant, the Brazilian variant, and also, unfortunately, there seems to be a very effective in terms of transmission a variant in India at the present time. So we have that inactivated vector protection in mice, and we also have the same with a live vector, and can get very effective protection in these different animal systems. So let me perhaps, we have all of these data, part of it has been published, and in summary, I want to say that the NDV hexaproline spike is effectively expressed on the surface of the Newcastle disease virus, and we have very, very good protection data in mice and in hamsters, and we are able to use both the inactivated as well as the live virus platform, which I think will be important for mucosal immunity in the future, but also we hope that there is a boosting capability if we have already a certain strain and variant, that by giving it one more of these variant vaccines, we should be expanding the immune response against different COVID-19 strains. And we don't know what is best in humans, whether the intranasal administration should be followed by intramuscular administration, or whether it should be the other way around. And we have also made quite a lot of these variants already. So this, again, I want to acknowledge it is a collaboration between the Kramer lab, the Garcia Sostre lab, and my own lab. And we have collaborations with Jason McLellan from the University of Texas. And we are in phase 1 trials in 5 different countries at this point. We are in Thailand, in Vietnam, in Brazil, in Mexico, and we also have a vaccine phase 1 trial going on at Mount Sinai. And this is acknowledgement also in terms of the support. We have PATH, which is the Melinda Gates Foundation, and we have also philanthropic donations from Mount Sinai, and also very effective support by the NIH. So this is really what I would like to stress in terms of the NDV-based SARS coronavirus vaccine. We have 2 papers which describe the live virus as well as the killed virus platform, and we hope that this will be in people, and through this humanitarian effort we'll be able to reach most of the population in our 8 or 9 billion people on our planet. So let me end here perhaps, and I just perhaps should just mention that something very strange is happening right now, that we don't have any influenza. And that is something very, very strange, which we don't understand. In the last 3 months, January, February, March, we had 100 times fewer cases of flu as compared to the same period 1 year ago. And we have 100-fold fewer hospitalizations and 100-fold fewer deaths. And we don't know whether this will be changing in the next year, whether we'll be harder hit next year, or whether flu is petering out. I think all bets are off. And I think we are in a very important time, which we don't know whether we will have either flu alone, COVID-19 alone, or both together, or whether they both, we would wish they would go both the way, but no one really knows. Let me take some of the questions which you might have.
Peter, that was spectacular. Great science. And yeah, this is such a critical point about what happens when we encounter our good old pathogens again. So audience, go ahead and either type your questions into the Q&A box, or you can raise your hand. So Jeffrey, can you go ahead and unmute yourself and ask your question?
Yes, I was just wondering, it sounds great in terms of the immunizing people against predicted threats. I was just wondering what the limit is on doing so. Can you only do so many a year, or is there a limit to the maximum number of targets that you can induce?
Are you talking about influenza or COVID-19?
Honestly, either, but more towards influenza, I think.
We would think that 3 stalks, there are the group 1 hemagglutinins, which are related, the group 2 hemagglutinins, and the influenza B virus hemagglutinins. We would hope that the 3 stalks would cover the entire sort of potpourri, the entire, all the 18 subtypes which we have in influenza A viruses. There may be a 19 or 20 in bats, etc. It ain't over until it's over, but we hope that with the current 3 stalks, we would be able to cover and neutralize all different strains belonging to all different kinds of influenza A viruses and B.
Thanks.
Peter, I had a more technical question related to Jeffrey's question. With what capsid proteins can you pseudotype Newcastle virus? Can it accommodate a great deal of things, or is it a subset?
Okay, so I went maybe too fast over it, but we anchor the SARS coronavirus 2 with a transmembrane region of Newcastle disease virus and the cytoplasmic tail of Newcastle disease virus F protein. So we fool the virus. On the outside of the virus, it is SARS coronavirus, but then the transmembrane region and the cytoplasmic tail tells NDV, yes, please package me. So I think there would be, by having this trick of fooling Newcastle disease virus to recognize it as one of its own, we should have quite a lot of different spike proteins, not only from SARS coronavirus 2, but from other viruses as well.
Yeah, I mean, surely there must be some constraints. I mean, even with the intracellular domain to recognize, even if there's like steric constraints, I can imagine that some capsid proteins would, maybe just be too big or have, wrong. I was just curious.
All of the above, yes, all of the above, yes. And more, let me tell you. Nothing.
Okay. I had a question about the silenced head domain. You said it was a head domain that we had not seen before. Did you guys just scramble a head domain, or?
No, we tried to use some other globular non-human blob. Yeah, we could not get this to work. So we had to use influenza virus hemagglutinin heads from very exotic animals.
Okay.
Okay, so these are the 8, for example, 18. They are in bats or in reindeer, in other animals which have not yet jumped over to us. You know? So that's sort of the exotic one, and we couldn't. We would have loved to scramble to the head, but we didn't. Yeah. We spent quite a lot of time to sort of give this a non-human tip, but it was really not possible to do that.
Yeah, but I'm curious, what was the failure mode for those? Did it not produce a good titer, or?
Yeah, I mean, it's very simple. If the damn thing grows, then the head is fine. Yeah. So yeah. we didn't have, it was very simple. We just put it into tissue culture or eggs, and if you get virus out, then it's fine. Yeah. But, okay. But there are quite a lot which didn't work. Yes, you're right. Okay. But we don't talk about those.
Yeah. And wait, so this is just my ignorance. So why would the novel head domain not be immunodominant?
Yeah, it would be. So we have to be careful not to give it twice. If you give it twice or 3 times, it's all over. So one has to be sure, and that's one of the problems. We have to, if you give 2 doses, it had to be, one is a red one and the other one is a black one, or whatever. You have to really, if a person gets twice red, then he or she will only make antibodies against the head. Absolutely, very good comment. Yeah, and that is a little bit of a problem. Yeah.
Yeah. Oh, that's fascinating. So you probably want like 3 dead versions to be able to.
We have actually 6 in each. Yeah. But again, it should never be the same one. Yeah. Twice.
Yeah. Oh, that's so interesting. I got a bunch more questions. One about the phylogeny and evolution of influenza. Why does influenza B not really seem to diversify as much as A?
Okay, very good point. That's what it is. It turns out that the A head is really a very demarcated domain. It has a disulfide bond on the bottom. The B virus doesn't have that. So when you make some changes in the stalk, then the B gets very nervous, the head. So it's probably some structural uniqueness of the A. It has the head domain and the stalk domain, and you can do a lot at the head and a lot in the stalk. But the B virus is more interconnected. So if you start futzing around with the head, the virus doesn't like that. But it's a good point, and we wondered about it, and that's what it is. Okay, A tolerates these different subtypes. B doesn't seem to tolerate that much.
Yeah. Again, this is my ignorance. Is that related to why B is usually less pathogenic than A, or is that some other aspect?
I think it should. Okay. It cannot make, or it does not make pandemic strains because it does not exchange the head, and therefore it is less virulent because there was no 1918 B virus in the last probably 300-400 years, and I think that's connected, yes.
Okay.
And not as much variation is possible.
Yeah. This is also interesting. Audience, do you guys have any questions? I've got another speculative one. So talking about this slide about what happens when we encounter our old friends again. So you spoke about it from the perspective of Bcells, about losing antibody response. But I mean, I know this is hard to predict, but do you think that T cells might come and save us, since maybe we'll have more populations of a bigger naive T cell repertoire to draw from?
Yeah. So there are B cell guys and T cell guys, okay, in flu. And this is 50, 70, whatever years we have B and T cells. So people are very in one camp or in the other camp. And it's yours truly is on the T cell camp. So I try to ignore T cells, you know. but there is an ongoing debate. If you would invite Rafi Ahmed, he would give you a different story, okay?
Yep, fair enough. All right, Peter, well, thank you so much for your time. This was really amazing. And I really hope that these vaccines make a big difference for developing countries. And then obviously we're really looking forward to the universal flu vaccine.
Good. Thank you so much. It was a pleasure on my part, and really very interesting discussion.
So join us next week for another great seminar. Okay, stay safe.